this wonderful video. So we're going to look at biology paper 2 revision, and I just have 2 questions. The first one is saying figure 1.0 show the structure of a living organism Rhizopus on the substrate. So we have different parts here. We have H, I, J, K, and also the substrate. So let's see the first question. So the first question is asking us to name the labeled parts H, J, and K. Okay, so what we have here is the spore case. So if you want, you can just say sporangulum, or you can say spore case. They are just the same. Or you can say the spore case. Then part I is called the sporangiopho, but we haven't thought to name this one. Then we have this one here, which is called the connecting stolon. So the connecting stolon is the one that connects to Rhizopus, 2 different Rhizopus. As you can see, this one is a different Rhizopus, as well as this one is a different Rhizopus. So there is a connection here. And that's the reason why it is called the connecting stolon. Connecting stolon. So that is what it does in terms of the function. Then okay, so let me just say not the sporangulum but spore case here, because the sporangulum is the entire part here, the entire part, and then these are spores. Okay, so those are the spores. Yeah, can just say spore. Then for K, this one is called rhizoid. Don't say the root. Of course, it is the root now. The specific name for the root of the Rhizopus is called the rhizoid. So the function anchoring and as well as releasing the enzyme or digestive enzyme on the substrate. Then explain the 2 functions of the part labeled K. So the function of the part labeled K as the Rhizopus 1, it anchors the Rhizopus, anchors the Rhizopus on the substrate, anchors the Rhizopus on the substrate. And that substrate can be anything, okay? Substrate. It can be the blade or the decaying organism. Substrate anchoring the Rhizopus on the substrate. Apart from that one, secreting digestive enzymes. Secreting digestive enzymes. So these digestive enzymes, they are going to digest starch into maltose, which is going to be absorbed by the Rhizopus. Okay. Alright, so the second one is saying name the type of nutrition demonstrated in figure 1.0. So that one is called saprophytic nutrition. So usually saprophytic organisms, they obtain nutrients from dead organism or dead living organisms. Okay, so we can say that one is going to be a saprophytic or saprophytic nutrition. And also holozoic nutrition that can take place in animals like us human beings. Then give the type of reproduction the Rhizopus undergo to produce young ones. The type of reproduction, it can be asexual reproduction, because those spores are going to be released from the sporangulum or the spore case, and then they are going to be implanted somewhere. So no need of fertilization here to take place as the meeting of the male and the female egg. So that one is asexual reproduction. Asexual reproduction. So what usually happens here is that once the spore lands on an area which is conducive for them to germinate, they will be able to germinate. So that is one thing that you need to know. Then we have this interesting question, which is saying a woman with blood group A, okay, and a man with an unknown blood group have 3 children with the following blood groups. So we have child 1 with blood group O, we have child 2 with blood group B, we have child 3 with blood group A. So as you can see, we already know the blood group of the mother or of the woman. So this one can be a homozygous dominant, or it can be a heterozygous dominant, whichever way. But according to the offsprings that we have here, we can say that A is coming from the mother. What about B? So B is coming from the father. So the father can be B, homozygous dominant, or B, heterozygous dominant. But as you can see, we also have a child who is O. So where is O coming from? So O is coming from the mother as well as from the father. So therefore, in terms of the genotype of the mother, we are going to have this one here and this one here. If you cross this one and this one, you're not going to have a child who is having blood group O. You can try. So the question is saying, deduce the most likely genotype of the mother and father. So for the father, since the blood group was unknown, so we are going to give him as B. Okay? And then for the mother, we are going to give her as blood group A, heterozygous dominant. So that when you cross, at least you are going to have all these offsprings are going to be found on the genetic diagram. So you can just try by using the Punnett square. So if we cross this one and this one, then we also have B and O. So of course here I'm not using the alleles for the sake of time. So when I cross here, I'm going to have a child who is having AB. Then here I'm going to have a child who is having blood group B. Here I'm going to have a child who is having blood group O. Let me say A. Then this one is going to have blood group O. So at least as you can see here, we have, according to the offspring, the genotypes that we have, blood group O is there, blood group B is here, blood group A is here, and additionally we have a child who is having blood group AB. So these are true genotypes. Okay. A man claims to be the father of the first child, but the laboratory results show that his blood group is AB. So the father would claim to say the first child, this is the first child who is having blood group what? O. Okay? Is our child. And this man is having blood group AB. Now using a genetic diagram, explain whether this claim is genetically possible. So at the end, we need to come up with a conclusion if it is genetically possible or not genetically possible. So remember, the mother is having blood group A, heterozygous dominant. So we can start with the phenotype. So phenotype. So under phenotype, what do we put? We put the mother or the woman close with the man. Okay. Then the next thing, we need to move on to genotype. So under genotype, we are going to use the alleles. For the woman, is having blood group A, heterozygous dominant. For the man, is having blood group AB. This one. Then splitting the gametes. So this one is going to be separate here. Even here, this one is going to be separate here. Even this one here. Even this one here. Then we cross them. So let me start with the first allele on this side here. So this one is going to be close with this one here. And then I'm going to have blood group A, homozygous dominant. Then also this one is going to be close with this one. Then I'm going to have a child who is having blood group AB. Then this one is going to be close with this one. Then I'm going to have a child who is having blood group A, heterozygous dominant. Then lastly, this one with this one, blood group B, heterozygous dominant. So as you can see at this stage, of course, you are supposed to put random assortment. Random assortment. Then after random assortment, this one is going to be F1 genotype. F1 genotype. So under F1 genotype, this is what we have. What about not F1, okay, yeah, F1 genotype. And then also this one is going to be F1 phenotype, where we are going to specify. So blood group is this one. So this is blood group A, and then this is blood group AB. But you're supposed to write it in full to say this is blood group A, this is blood group B, and this is blood group A, and this is blood group B. So as you can see, are we having any child here who is having blood group O? Because the first child here is blood group O. Now, are we having any child here who is having blood group O? No. So meaning to say, the claim was not genetically possible. So the conclusion is going to be, the claim was not genetically possible. So this is how you're supposed to conclude, as simple as that. But if you're not going to conclude, meaning to say, they're not going to mark you. Okay, so this is how you're supposed to conclude. So the claim was not genetically possible, then you are done. They're going to give you 5 marks. So thank you very much for watching, and remember to subscribe for more videos like this.